The overall goal of this procedure is to diagnose plant virus diseases within a few hours by transmission electron microscopy. This procedure begins with microwave-assisted fixation and embedding of the infected plant material. The next step is to extract and stain virus particles from the plant samples by negative staining. Followed by sectioning of the embedded plant samples. The samples are then investigated in the transmission electron microscope, and the size of virus particles is measured by image analysis. Ultimately, the results show typical virus-induced ultrastructural alterations and the size of virus particles through transmission electron microscopy methods.
The main advantage of this technique over existing methods such as conventional fixation and embedding at room temperature, which can take several days, is that sample preparation time for transmission electron microscopy can be reduced to about 2 hours, thus allowing a clear diagnosis of the viral disease in about half a day. Demonstrating this procedure together with me will be Gerhard Graggaber, a technician, and Professor Günther Zellnig, a colleague from our laboratory.
Before the start of sample preparation, program the automatic microwave tissue processor for electron microscopy, Leica EM-AMW, with the appropriate protocols. Prepare the solutions for the different steps described in the programmed protocol for fixation, dehydration, and infiltration.